All right, so welcome to your lesson on the nature of light in photosynthesis. Now, here's the thing about light. We don't really know what light is. You will find disagreements all over the place in the physics world because in some ways it behaves like a wave, similar to what you would see in the ocean or hear a sound. And in other ways, it doesn't act like a wave at all. It acts like a particle. It acts like a package of energy called a photon, or the fancy word is quanta. So if you're studying quantum physics, this is your realm. So it kind of behaves in both ways. So when we're discussing light, sometimes we talk about it as wavelengths, sometimes we talk about it as photons. In both cases, what we're talking about, though, is specific size of that wave. So if it's a photon of light, it's at a specific wavelength. Okay? Different wavelengths have different characteristics. When we look at the overall electromagnetic spectrum, every single one of these types of waves comes out of the sun. They're all available there. They're coming and hurtling towards Earth at ridiculously high speeds. The radio waves are the very largest ones. And just like some membranes can be permeable to certain things, or objects, we can describe them as being transparent to light, like a piece of glass is transparent to visible light, so is literally every other object. They're transparent to something, and they are opaque to other things. So a radio wave right now is passing through this room and passing through your body, and you have no idea about it. The only way that a wave can actually interact with you is if it affects your electrons and causes them at just the exact perfect wavelength, causes them to get excited and jump up to a new orbit level. So if I can get that electron excited and moving, then I'm going to have... that's what we call something called fluorescence. I can excite it, it moves to a new energy level, and then it will lose that energy and drop back down and fluoresce that energy as some kind of light. It converts it in that way. Radio waves cannot interact with you. Microwaves are very good at interacting with water molecules. That's why when you throw your food in the microwave, really what you're doing is vibrating and adding energy to the water in your food, and that water, once it absorbs those microwaves, it converts that into heat energy for you and warms up all the particles beside it. That's how microwaves work. Infrared, again, is a different... we can use infrared to cook things because now we're, again, treating things on a different scale. And then you have the visible light, which is what our eyes can detect. So we have that fancy little retina on the back with all the rods and the cones that allow us to see light. UV we can't detect, but there are certain types of bugs or birds that are able to see in the UV spectrum because they have special little cones that can just on the edge of that. But UV, I think you know, is what tans your skin. Did you know it also tans your eyeballs? Whereas your skin will produce more melanin and become darker, no matter what background you have. It doesn't matter how dark your skin is to begin. You will always get darker if you spend a lot of time in the sun. And then your eyeballs actually get lighter in the summer. Pay attention this summer and you'll see if it might be ever so slight, and maybe you don't spend enough time outside, but if you're spending enough time outside to bleach your hair and to brown your skin, you're probably lightening your eyes too. So it's pretty neat. UV is able to do that because it has the right wavelength to interact with your cells. But I think you also know if you spend too much time in the sun, you can also cause skin cancer. And that's because UV, even though it can only penetrate it can't go all the way down to your bones, but it can penetrate just the top layer of your skin. If it happens to interact with DNA molecules and it damages them, then it can actually, that switch that's supposed to tell a cell you're only allowed to divide so many times before you die, that gets broken. And then the cell actually changes and it begins to grow and divide out of control. And it's no longer sticky to other cells the way it should be, and all sorts of characteristics change because the UV has damaged the DNA on-off switch. Yeah, so when you wear an SPF sunscreen, what you're trying to do is, because there's only a very thin depth that the UV can go to, what you're doing is creating an extra barrier so that the UV is absorbed into that stuff rather than going into your skin. Now, they generally sell, most of the time you'll see SPF 60 out there. Beyond SPF 15, it really doesn't add any extra protection. So save yourself some money and buy the cheap stuff, because 60 doesn't do any much more. What might be nice is if it has, like some kind of a moisturizer in it or something, then great. But other than that, beyond 15 doesn't do a whole lot more. 60 doesn't do a whole lot more than 15 does. But it sounds better, so people buy it. So UV can penetrate skin and can cause damage. X-rays can penetrate even farther. you know that X-rays are what we use. They pass right through the body. They do get slightly absorbed by more dense materials like your bones, but they'll pass very nicely through soft tissue like your skin and your muscles. And so we can see a different set, very dense things we can see. So we can look for tumors with X-rays. We can look for broken bones because it tends to be more dense, so it'll absorb those particular things, right? Now, gamma rays, I'm sure you've heard of that documentary, The Hulk. He's been in quite a few documentaries. I believe the series of documentaries is called Avengers. And you know what gamma rays do to people then if you've ever seen The Hulk, right? True story. Okay, gamma rays, because of their high intensity and because of who they can interact with, especially like gamma rays are the worst, X-rays are pretty bad. If you're exposed to a lot of X-rays for a long time, you can lead to cell damage. If you're exposed to gamma rays for a short period of time, it can lead to cell damage and to cancers and all sorts of stuff. So when you've got let's say a nuclear fallout from a nuclear plant exploding like Chernobyl or something like that, or even the bombs in Hiroshima and in Pearl Harbor, all of those radiation being thrown out was damaging the things around. Actually, it's very interesting if you go and you study and you look at the mutations that have happened to the animals and the plants in those particular areas. Some really interesting things have happened to the animals that are there, but that's for another time. So what we are dealing with when we're talking about plants is within the visible spectrum like you saw in that other diagram. We're going to be absorbing a lot of waves in this particular area. We're going to be releasing and rejecting the ones in this particular area, and then a little bit over here in the orangey red. But let's talk now about the actual fluorescence and how normal fluorescence differs from photosynthetic fluorescence. So we've already discussed then that when a molecule gets struck by a wave or a photon of light, however you want to describe the light, it's at a certain wavelength that it can actually interact with its electrons. If it's any other wavelength, that molecule will not be affected. It'll be it'll just be transparent to that particular set of wavelengths. Just like I told you, the radio waves will pass right through your body. They're not going to make contact with your cells. They'll pass right through you like you're transparent. If they do, if it is the perfect wavelength and it can interact with that molecule, then we would say that the electron then, in general, will fluoresce. And the idea would be that when the photon of light strikes it, it gets excited and jumps up to another energy level, but it won't stay there forever. And when it jumps back down to normal, it'll let go of the energy it got from the light, and it'll release it as light or as heat. And so then we would say at this point in time, when the electron returns to its ground state is where it started, then we would say it fluoresces, just releasing that energy. So you get excited, it jumps up, and you release that energy. This is the same thing that's happening when you explode fireworks. You have different types of metal that are going to fluoresce at different wavelengths of light, producing different colors. So if you take, let's say you take a sparkler, let's not even talk fireworks, just talk just sparkler, that's made of magnesium. When you light magnesium on fire, it burns this bright white. And what's actually happening is you've heated up the magnesium, the electrons in the magnesium have gotten so excited that they've done this and they've jumped up, and then as they jump down from that energy being added, they let go of white light. If you do that with lithium metal, you get this beautiful red color. Did you guys get to do flame tests in any of your other courses? It's quite fun. Sodium does this beautiful yellow. Copper does this wonderful like green color. Do yourself a favor. Next time you go camping, just take a little tiny piece of copper tube with you, like what you'd have for your for the plumbing in your house. Little tiny piece of copper tube. Throw it in the campfire. You'll get these beautiful green colors coming out of your campfire. And you can just keep using it over and over again because you can always just rub off the oxidation of the copper and it just keeps going. Point being, it's going to jump up, get excited, and when it drops back down, that whatever package of energy it throws back out, that photon of light it throws away, will be at a certain wavelength, and you'll get the different wavelengths of color popping out. It's really quite cool. That's normal fluorescence. What's different about photosynthetic fluorescence is that you're not just dealing with any molecule, you're dealing with chlorophyll. And chlorophyll has this weird thing that it can do, different from other molecules, specifically chlorophyll A, in that when you excite it and the electron jumps up to the new energy level, rather than it jumping back down, right next door there's another molecule that's waiting to steal that electron. And that is the major difference, is that you're not just allowing the electron to bounce back down again, you're stealing it. It's too far away from the nucleus, and I'm able to rob that nucleus. So in essence, what I'm doing is oxidizing the chlorophyll. I'm making it a positive charge. It's no longer neutral. On one of our first lessons, I wanted you guys to look at reduction versus oxidation, and this is where it comes into play. If I give something an electron, I have reduced that thing, but I gave up my own electron, so I'm oxidized, and the guy who got my electron is now reduced. And vice versa. If that guy gives up an electron now back to me, it becomes oxidized, and I become reduced. Okay? Oxidation reduction. And that's where I gave you that LEOGER, L-E-O-G-E-R, losing electrons oxidation, gaining electrons reduction, or oil rig. Oxidation is losing, RIG, reduction is gaining. So you have to keep those in mind. Together, the idea of one losing and one gaining the electron, we call that a redox reaction, because both reduction and oxidation have to happen at the same time. They always happen at the same time. If somebody gains, somebody has to have lost. Okay, so redox reactions. So that electron then gets lost from that chlorophyll molecule, and if I want the process to continue, I have to reduce the chlorophyll again. I have to give it back another electron. And the way that I do that is I steal it from water. So we're going to go through a process called photolysis, photolysis, photo meaning light, lysis means to burst or break apart. So what we're doing is taking advantage of the fact that light has already struck this huge big molecule, and it's going to cause not just the electrons to jump away from the chlorophyll, but it's going to activate another enzyme, and that enzyme is going to grab water and burst it apart into 3 parts. We get our oxygen. We get the hydrogen split into its 2 halves, so we get it split into its protons, represented by the H+, and its electrons, represented by the E negatives. So these electrons that I steal from water will then be given back to this chlorophyll molecule so that it can be oxidized again. Keep the process going. Okay. And then the electron that I did steal, it doesn't just stay with this one electron acceptor, because the problem is that you've got all these acceptors all in a row, and each one of them is a greedier electron hog than the first guy. So it's basically like the mafia organization. You've got the mob boss, and you've got the guys under him, and then you've got like the minions, right? So this is more like a minion. He's just stole the lunch money from the guy around the corner, and then he's got to give the lunch money to his boss, who gives it to his boss, who gives it to his boss. And then the big bad dude decides what to do with that electron. And the basic idea of passing an electron is that we are passing energy. It's a form of energy. So again, I'm going to take that electron and I'm going to then pass it through the chain, and you'll see the chain in our next lesson. Here we are again looking at the different plant pigments. So we've got our main chlorophyll A, which you've already said is our primary pigment. We have B and the keratinoids, which we're going to call accessory pigments. There's lots of different types of keratinoids. Just throwing a couple out at you. If you're thinking about the orange color, you would say beta carotene. Again, that's the exact same vitamin that we see in oranges. This is the beta carotene, is the one that they pretend that if you eat a lot of carrots, it's really good for your eyes. Well, it's true actually, because of the beta carotene. It's a very good pigment for your eyes. And then xanthophyll happens to be the yellow color. There are other ones listed here. Erythro is what we call, if you remember back to red blood cells, they're actually called erythrocytes because they're red cells. Erythro means red. So this is just something that would be in the red range, and then the cyanin, the phycocyanin, would be in the bluish kind of a range. So it would be what it would be giving off, right? So remember that this is an absorption spectrum. This is the wavelength of light that they like to keep. Anything that isn't along their line, you would be reflecting. So again, when we say chlorophyll A is absorbing in this wavelength, it's not absorbing here, which means that's why it appears that greenish kind of a color. Same thing with chlorophyll B. It'll release the greenish section, whereas the keratinoids are going to release more of the reddish kind of an area. Plus, you get all these other guys here as well. So this one is absorbing within the red spectrum, and this one's absorbing within the green. So it would not appear those colors. It would appear the opposite of that color, and so on. Right? So that's why we're going to get the beta carotene appearing orange, because it reflects the reddish yellows, but it absorbs the other things. Okay. This is also why leaves change color in the fall. Leaves are always orange and yellow. or red or whatever it is that they change to. They are always that color. These pigments are always there. But during the warmer months, you have so much chlorophyll, which is green, it overwhelms the other paint that's there. And so until you come into the fall when that chlorophyll begins to break down and be recycled, then all of a sudden you're actually able to see the carotene that's been there the whole time and the xanthophyll that's been there the whole time. So that's why we get the fall changing colors of leaves. Okay. Now, why do we care about carotenoids? This is definitely the test type of a question. So that's why it's starred on the side here. What do carotenoids do? First thing that they're going to do is absorb any of that light wave that would actually possibly cause damage to the chlorophyll. They're going to absorb that themselves, and then they're not going to pass it on. Instead, they're going to fluoresce it, and they fluoresce it as heat. They get rid of it. Okay? So any harmful wavelengths, they will fluoresce. If it is the right wavelength of light, though, something that would benefit the chlorophyll, then it absorbs it and passes it on. So it will channel the light energy to the reaction center. And again, the reaction center is always a chlorophyll A because chlorophyll A is the only one that actually gives away the electron. So carotenoids are going to absorb and fluoresce light that would otherwise be harmful to chlorophyll. And then they're also going to absorb helpful wavelengths of light and pass it to chlorophyll. And this is what we're talking about when we're saying that we're going to be passing the energy. Here we have a structure called the Photocenter. It's literally set up like an antenna array. So if you think about what your satellite dish looks like, this looks like a satellite dish. And in the very center, you're going to have your chlorophyll that's reactive. That's your chlorophyll A. And all the way spread around it in 3D, you're going to get all these different pigments. When a photon of light comes and strikes the pigment, if it's harmful, it just fluoresces it, gets rid of it as heat. If it's a good package of light, it'll pass it and pass it and pass it and pass it until eventually that energy gets to the chlorophyll. Then it can lose its electrons. So everybody's fluorescing and dropping down, fluorescing and dropping down, fluorescing and dropping down. And this final guy will fluoresce, an electron will be stolen. And that's how you start the process of photosynthesis. Just get the energy to the right spot. This particular chlorophyll, there are two different types. You'll see them in the photosystems in our next conversation. But they specifically like and prefer either the wavelength 680 nanometers or 700 nanometers. And you'll see how that comes into play later. So they're very, very specific on which perfect part of light they want. That'll be absorbed by that perfect electron that will then cause that exact electron to jump and be stolen. And then here the chlorophyll gets oxidized and the electron acceptor gets reduced. But that's not the end of the story for the electron. Once I have excited the electron, it passes to another particle, which passes to the B6F, which passes to the plastocyanin, passes to the photosystem 1, gets re-excited, passes to ferredoxin, passes to NADP reductase, and then the electron finally ends up in this molecule here, which then floats off to go and help us do more chemical reactions. So I'm just bouncing from structure to structure to structure to structure to structure all the way through the membrane and then eventually giving up that electron to a molecule that can float away. Then I have to replace the original electron I lost, and I do that by splitting the water apart. But this is a conversation we'll have over the next couple of lessons to give you more of those details. In this particular version of our thing, we've also included the states of matter. So it would be a gas, a liquid version of water, but then we would have an aqueous version of glucose, the AQ. Just want to remind you that just means glucose is floating around in water, which makes sense because I'm going to have to transport this glucose through the phloem tubes. So I want to make sure that I'm able to do that. And water is one of the best ways to move things around. And then, of course, the oxygen also would be gas at the end. These next couple pages are just there to try to give you an overview of what's coming. We're going to get into lots of details in the next couple of lessons, but this is your basic overview. The most use you'll get out of this is when we've already finished the detailed lessons and you're coming back with understanding to see, oh, this is all of it in one place. But I do still want to just go over it with you while we're still walking through it. So again, keep in mind, if you want to do well on this particular unit, whether it be the photosynthesis side or the cellular respiration side, you want to follow the carbons and you want to follow what happens with the water and the oxygen. Always following the carbons, okay? So we want to look at the major pieces of each of this big reaction here and say, where do they come into play? Like specifically where? Am I in the thylakoid? Am I in the stroma? Who's using it? How do I actually use it? What role does it play? So water, think photolysis. I'm going to blow it apart. I'm going to steal some electrons. I'm going to have some protons that'll do some work for me, and then that oxygen is going to be released. Okay, carbon dioxide, you'll see in the second stage, we're going to be fixing it to some molecules, sticking it onto them, and eventually we're going to end up with a half sugar that'll eventually become glucose. So you want to know where and when those things are happening, and what conditions do I need to make it happen. Mentioned this before, I'll say it again. Glucose is the most common sugar we're going to produce, but it's not the only sugar. So when we say glucose every time, it's because we're simplifying the conversation. And again, the oxygen that you see there is coming from splitting water, photolysis of water. It does not come directly from the CO2. So in general, here's what's happening. Here we have a simplified diagram of the chloroplast. You got the inner and the outer membrane. And then you got your thylakoid disc in a granum, in a big stack. And built into this membrane, you're going to see the chlorophyll has been embedded, and then a whole bunch of molecules have been embedded as well. But in general, the light strikes the chlorophyll, that excites an electron. The electron starts bouncing through this chain. Water gets split to replace it. And through this process of moving protons around, and I'm going to end up with these two important molecules, an ATP and this NADPH. NADPH is carrying a high energy electron on the end here, disguised as a hydrogen. And the ATP is also a high energy molecule. These guys are going to be now the energy, the chemical energy that powers the fixation of carbon dioxide. When we say carbon is being fixed, we mean it's being attached to a molecule. It's being built in. It's being fixed in place. So this is going to drive these reactions, and this reaction called the Calvin cycle is going to produce our half sugars for us. And then half sugars get put together into full sugars and so on and so forth. So that's it in general. I'm going to need to, once I pop the phosphate off of here and steal this electron, then the main part of it, the ADP now, because ATP is 3 phosphates, ADP is diphosphate, 2 phosphates. Then I just go back and I pick up another phosphate and I deliver it. And I pick up another phosphate and I deliver it. And here I've lost electrons. So I go back here, I get reduced, take those electrons to the next stage. I give up the electrons. I go back and I pick up more electrons. So these guys are going to be the vehicles that just constantly give us what we need to run this reaction. So that's in general how we're getting light energy to suddenly become chemical energy. We're just stealing and exciting electrons, and it's the electrons themselves that are able to then cause these reactions to happen. Okay. So when we look at our two main stages, we have something called the light-dependent reaction, because it requires light. And then we have the light-independent reaction, because light has nothing to do with this. This reaction is sometimes also called the dark reactions. So you have the photosynthetic light reactions and the dark reactions, but this got confusing for people because they thought that dark meant it had to be at night, and that's just silly. Dark just means you don't require light. It doesn't mean it has to be dark. It just means light is not a part of our process. So middle of the day, you can have light-independent reactions happening. The other name for it is Calvin-Benson, named after the two people that were both given credit for having worked on it. Calvin-Benson cycle. Or usually Calvin is the main person, so Calvin gets most of the credit. Benson kind of came along afterwards and gets the credit afterwards. But so Calvin-Benson or Calvin cycle or dark cycle or light independent reactions, because why have one name when you can have 12? Okay. So the other thing you want to do is not just compartmentalize and say what happens in each stage, but where does it happen? So this is happening in the thylakoid membrane. This will be our next major lesson. And we're going to grab that light energy from those photons, helps us to oxidize the chlorophyll, meaning we're stealing the electron. The electrons bounce through this chain of molecules. That's going to cause something called a chemiosmotic generation of ATP. I'll show you all that in the next lesson. Basically, what we're doing is moving protons, and that drives a motor that crunches and creates this ATP. The electrons themselves end up in NADPH, and again, these are the two energy molecules we're going to use to drive these reactions down here. You have to have chlorophyll A, you have to have photons, and you have to have the building blocks for these guys to get things going here. And you have to have water in order to replace the electrons that you're going to lose from chlorophyll. Light independent happens outside the thylakoid in that juicy bit in the stroma. And again, we're going to use that ATP energy. We're going to use protons that we had from before, and we're going to fix that carbon. All right, so what are we looking at here? Well, this is our first stage. This will be your first major next lesson. looking at the light-dependent reactions, so we know these photons of light will be involved. In the first half of it, we're going to take that water and we're going to split it so that we can replace the electrons we just lost from the chlorophyll. Then we're going to care about these products that we're going to make. We're going to make some ATP and some NADPH. I'll show exactly how we do that. What we really end up doing is taking a phosphate. I know it says capital P, and you're thinking phosphorus, but in this particular thing, it's actually a phosphate, a PO4. It's simplified as a P all over the place. And so they usually put this little I in there to indicate it's an inorganic phosphate. It's not just a phosphorus atom. Okay? So ADP gets this extra phosphate group attached to it. So it goes from being diphosphate, 2 phosphates, to having 3 of them. And this is your general currency for cellular energy. This will be the goal of cellular respiration as well, is to give us a whole ton of these. So the plant is just doing this as an intermediate step to, it's going to use it here, and then it's going to take that energy to drive the next reaction. It's going to do the same thing with the NADPH that you get. The electrons that you just stole from the chlorophyll end up causing this positively charged, this should say NADPH. I didn't catch that before. Hang on. Apologies for that. NADP. So this, I guess we could say it's an oxidized version of it because it's already lost an electron. It gets crunched together with this proton, and those electrons are the glue that holds it together. There's a really important concept that you need to understand. Chemists explain it different from biologists. If I give you a hydrogen atom, okay, it has 2 parts to it. You have a proton and you have an electron. If I give you a gift in the bag, unless you are a cat, what's the most exciting part of that gift? Is it the bag itself, or is it the gift that's inside? Hopefully, again, unless you're a cat, you think that the gift inside is the better part, right? So the same thing here. If I give you a hydrogen atom or a proton plus an electron, the proton is not the gift. The electron is the gift. It's the main thing. So when you see something go from this form to suddenly have a hydrogen on there, I know it sure looks like you just gave them a hydrogen, but what you really gave them was the electron that glued these pieces together. That's the important part. So we would then say that the NADPH is an electron carrier. So it's going to take the electrons from that photosynthetic membrane out to where we've got some chemical reactions, and it's going to dump off that electron and allow us to rearrange some molecules using the energy from that electron. So anytime you see an H suddenly appear on a molecule, the gift is not the H, the gift is the electron. Okay? Stop playing with the bag, it's the electron. Now we call this a reducing agent. And this is where the terminology sometimes gets annoying, because if you've got the idea of oxidation reduction down, you think you've got it all. Okay, oxidize, I've lost electrons, I become positive. And then reduced, I have gained electrons and either become neutral or negative. Great. But what the heck is a reducing agent? Well, if I spin it around and I call it an agent of reduction, then hopefully you get the idea that this is a thing, an agent, who is doing the job of reducing something else. So a reducing agent reduces something else. How does it do that? It does that by giving it an electron. So it carries the electron right now, but it will reduce something else shortly and give that electron away to another molecule in this stage down here for the light independent. So an agent of reduction or a reducing agent causes something to be reduced. An oxidizing agent would be stealing electrons, causing something else to be oxidized, even though it itself would be the opposite. So this guy, by being a reducing agent, actually becomes oxidized itself, while it reduces the other thing. It can be a bit confusing, but hopefully you'll get that. Now this is where we're going to get into these big long words that are describing the molecules that we're dealing with. These are just the organic terms. Once you get into organic chemistry, they actually don't seem so scary anymore. So in our second half of our photosynthetic stage, we're now going to deal with the carbon dioxide. This is where it comes into play. And through a bunch of processes in the Calvin cycle, we're going to use the energy that we created or stole in the first section, and we're going to use it to allow the carbon dioxide to get stuck or fixed to a molecule, run through the Calvin cycle, And then our end goal will be to produce this molecule right here. Actually, a whole bunch of this molecule right here. What is this? Well, P-gal or G3P is the exact same molecule. It's a carbon molecule. We haven't bothered to put all the functional groups on this, so it has 3 carbons to it and a phosphate group that's attached. And just the way that the functional groups are arranged around the carbons, again, we haven't shown them here, makes it a glyceraldehyde. It's just the way that it's constructed. So you can either call it phosphoglyceraldehyde because it has a phosphate attached to a glyceraldehyde, or you can be more specific and say exactly where is that phosphate, and you can say, well, it's a glyceraldehyde, and on the 3rd carbon, we've got a phosphate. That's the only difference between those names. It's whether you are giving the general term, it's a phosphoglyceraldehyde, or whether you're being very specific of the location of that phosphate. That's how organic chemistry kind of works. Now, it's only 3 carbons. We need 6 to make a glucose. So we will pause our conversation on photosynthesis at this point and just tell you that after this is created, there's a whole bunch of steps that are going to turn the 2 of these P-gals, or 2 G3Ps, into 1 glucose, so I have enough carbons for C6H12O6. Last little bit, simple concept. I hope the graphs are going to make sense for you. Here's the 3 things that we care about that are going to affect the rate of photosynthesis. The first one is how bright the light is. We call that light intensity. Low light versus high light. So low intensity versus high intensity light. How many photons are you actually throwing at the chlorophyll? Then carbon dioxide levels are going to matter because the more carbon dioxide I have available, that means that I should be able to be running that Calvin cycle over and over and over and over and over again, keeping all the enzymes and coenzymes busy. And then temperature is going to matter as well. Every reaction will increase with temperature because you have a higher random collision of molecules, plus you've got these enzymes doing their job. It just allows everybody to speed up the process. But you already know from looking at just enzymes in general that you can only heat up and increase to a certain point, and then it gets too hot and you actually break the machine. And if you break the machine, you're going to have a rapid decay in the rate of photosynthesis because you've broken and busted all the enzymes that are supposed to be doing the job. This one should make good sense, right? Temperature increases until it gets too hot and you break everything. These ones, I hope the graph makes a little bit more sense. Ignore the top 2 branches and just look at this branch for 1 second. What we're saying is that you will see an increase in the intensity of light, but it's temperature sensitive. So if it's cooler, you're only going to get so much intensity. It'll increase until it hits a temperature wall. And same thing here. It's even better. Increase, but then the temperature begins to affect it. And here as well. So if you can get it as much light as possible, and you have the temperature tweaked to just the highest it can go without being too hot, you're going to have very fast production of sugars. It's also, though, sensitive to how much of the substrate you have. If you don't have a lot of the carbon dioxide available, then you can have all the light intensity you want at the perfect temperature, but it will be limited how fast you can do it by how much carbon dioxide you have available to actually make the sugar. So if you have up with light intensity, but low, you're going to hit a wall. Up, but high concentration, you're going to eventually run out of it. You're still going to hit a wall when you run out of substrate. Does that make good sense? Those are the factors that affect the rate of photosynthesis. Please memorize light, CO2, temperature. 3 factors that affect. And they're all increasing to a point. And that, my friends, is the end of your first lesson on photosynthesis.